Thanks very much, Alex, for that introduction. It's great to be here today, and I'm going to be talking about, as Alex mentioned, systematic online living evidence summaries, or SOLES projects. And this is where we can use AI to help accelerate in vivo synthesis. So the outline of my talk today, I'm going to give a bit of background, but that's really helpful that Alex has already introduced me in this way because I won't have to spend very long on that slide. Then I'm going to talk a little bit about preclinical systematic reviews and why they're useful. I'm going to talk about the SOLES approach. I'm going to give you a demonstration of one of our SOLES projects. I'll talk about the future work that we want to do in this area, and then we can have a wider discussion and I can take some questions. So as Alex mentioned, I'm a postdoc at the University of Edinburgh working with the CAMARADES group. And I've worked primarily on systematic reviews and meta-analyses in the Alzheimer's disease domain, but also in other spaces as well, and mainly in animal research. I've also worked on automation tools to help support evidence synthesis, and I lead the SOLES development team. So I wanted to start with this broad statement that I'm hoping that everyone that's joined the call will agree with to some extent, that scientific advancement should proceed based on what we already know. And this is true for whether you're working in clinical research, whether you're working in animal research or cell-based research. We want to make sure that the next experiment is planned with a consideration of existing knowledge. The difficulty is that figuring out what we already know is not that simple, and that's why we need tools like systematic reviews and meta-analyses. So systematic reviews of preclinical research specifically allow us to help investigate the differences that we see between the preclinical evidence and the clinical trials. We know that in a lot of fields, drugs that seem to work in animal or cell-based models, when you bring them into the clinic to try on patients, they don't work in the same way. And we want to investigate why that is. We can also use a meta-analysis to explore the variability of results. So in certain fields, we might see that even within the same animal model, there's a lot of variation in the results that we see, and we can investigate that using statistical approaches by combining all of that data together. We can also use systematic reviews to inform future preclinical studies and identify where there's gaps in the literature. This also can play into 3Rs decisions because, for example, we can do an analysis of different outcome measures, and we might determine that a certain outcome measure is actually just as sensitive as another outcome measure, but might be more or less harmful for the animal or less invasive in some way. So it might help us select which outcome measures to use. It might also help us select which animal models to use under different conditions. They can also be used to help inform early phase clinical trials because they can help us determine if there's actually enough preclinical evidence in cells and animal-based models to take a drug forward into clinical trial. This also plays into reducing research waste because we might find if we do a systematic review that actually there's enough evidence out there and we don't need to do another animal study to confirm a finding. Overall, in the preclinical literature, we know that there's a huge amount of evidence being generated from in vivo and in vitro models. But there are actually few coordinated efforts to collect all of this data, all of this published data, to summarize it and utilize it going forward so that we can make evidence-based decisions. So hopefully I've convinced you, at least partially, that systematic reviews are useful, but there's a number of problems with them at the moment, especially using the traditional approaches that we would take to doing a systematic review. So the first problem is that they take a huge amount of effort and resources. So in the preclinical space, especially, in one of our previous projects that was looking at animal models of neuropathic pain, we found that it took 18 person months to screen the title and abstracts of 30,000 articles that we retrieved in our systematic search. That is a huge amount of time just spent reading the title and abstracts of articles and determining whether they're relevant. There's also been some examples from Cochrane systematic reviews. So Cochrane is, they perform systematic reviews in the clinical space. And actually, they tend to be a lot smaller than systematic reviews of animal and cell-based research. but there's been examples showing that over 50% of reviews are actually published more than 2 years after the original protocol, and 85% of those reviews are not updated for over 5 years. And the reasons behind this is that, first of all, just getting through the number of studies, screening them, annotating them, making sense of all that data takes time, and actually going back to a review to update it is not trivial, and sometimes just as much work as starting a new review from scratch. There's really a need for more scalable approaches, and in our research group in CAMARADES, we've been trying to do reviews that are broader and broader in scope. So right back at the very beginning, I don't know how many years ago now, a long time ago, back in 2005 or 2004, people in the group started doing reviews on very specific drugs in animal models of stroke. So they might have looked at one stroke drug in a specific animal model, and maybe they retrieved a few hundred results that they could easily get through within a short space of time and end up with an output. These days, we're trying to maybe answer bigger questions. So maybe we want to collect all of the evidence on animal models of neuropathic pain, for example, and get an idea of what models are useful in different situations and what treatments seem to work the best in those models. To do something of that scale, we need a different approach, because taking a manual approach is just becoming impossible. And this is compounded by the fact that the amount of publications containing animal research is skyrocketing over the last decade. So we just can't keep up with the pace of evidence generation in this space. The second limitation of current approaches is that they can actually fail to capture all of the available evidence. And we found this in a few of our reviews. So if you go into PubMed and you search for a specific animal model, for example, or a specific outcome measure, you will find that a lot of the relevant articles will not come up. So I found, for example, that 70 to 80% of papers in a review of the Alzheimer's disease literature in animal models didn't mention the outcome measure that I was interested in within the title, abstract, or the keywords. And those are the things that are indexed in PubMed. So if you're looking for all of the studies measuring outcome, measuring behavior in the open field test, for example, you're not going to find all of that using a traditional search. We've also shown similar things in the in vitro literature, that the model is quite often not mentioned in the title and abstract, and 14% of relevant studies were excluded for this reason. The third problem is that there's a lack of coordination. So sometimes there's multiple systematic reviews going on that overlap with each other. There could be different groups doing actually very similar systematic reviews. And there's just generally a lack of coordination in this field as a whole, in terms of the methodology used. And I think there's a real opportunity to try and facilitate a bit of coordination to do things faster and better with more collaboration. So as a reminder of the systematic review process, so we have to start off with identifying a research question of interest. We write a protocol before we begin actually working on that review. We identify all of the relevant literature that we're interested in, and then we screen the studies for inclusion or relevance. Once we have a set of relevant studies, we then would extract information from those included studies around the study design, so the animal model, the interventions that were given, we also extract information about the study quality, so whether they've taken measures to reduce the risk of bias, and extract numerical data from tables and figures if we want to do a meta-analysis. And then finally, we would combine all of that data for a qualitative synthesis and possibly a meta-analysis as well, summarising the evidence on that research question. So what we've been interested in over the last few years is how we can actually make this process faster and introduce automation approaches at each step. And I'm going to talk about this in a bit more detail, but essentially it means using machines to help automate some of those more manual tasks like searching databases and screening articles for inclusion. We now, over the years, have actually accumulated what I would call a sort of automation toolbox to help us, tools to help us with each stage of the systematic review. So for systematic searches, There are now APIs or application programming interfaces to a lot of the major search databases such as PubMed, Web of Science, and Scopus. And this allows us to actually automate the search process so that we can actually just have a script running that searches PubMed, for example, and retrieves the results on a weekly basis, rather than having to manually enter our search terms and find relevant studies. For deduplication, there are lots of tools out there now to assist with the problem of multiple studies, basically the same study being in PubMed and Web of Science and Scopus and elsewhere. There are tools to help us remove any duplicates in an automated fashion. There are also tools to help us screen studies in an automated way, which is using machine learning approaches based on the title and abstract of studies. We can actually train a classifier to screen those studies and determine which are relevant and which are not. We can use tools to extract information about the animal model and the treatments, the outcomes that are measured, and we can also start to get an idea of the reporting quality. So there are tools out there to extract, for example, whether animals were randomized to experimental groups or not. And finally, there are tools out there such as SOLES, which are meant to help with the dissemination of all of this evidence. So rather than the more traditional approaches of writing up a systematic review, getting it published, maybe doing an update 5 years down the line, we're looking at ways to try and keep this process up to date. And those are things like living systematic reviews, and there's platforms out there now to support that sort of publication, or I guess they go hand in hand, they go together, but also things like SOLES, which I'm going to talk about, which are these living evidence platforms summarizing all of the evidence in a given research area. So because of these issues, we've been looking to improve evidence synthesis, and we were looking to methodologies such as living systematic reviews, which are kept up to date through a combination of automation approaches and crowdsourcing. So using large numbers of people, volunteers, other researchers to help keep up to date with a systematic review. And we're also interested in evidence gap maps. So this is a methodology used at a very high level to get an overall picture of a research field. So they typically map out the number of studies looking at different things. For example, the number of studies that are looking at a specific drug or a specific animal model. So we sort of took a combination of these approaches and developed systematic online living evidence summaries, or SOLES projects. And these are really high-level summaries of an existing research domain, and it's visualized in an interactive web application. So the idea is that all of the evidence in a given space, for example, all of the research in animal models of Alzheimer's disease, for example, we pull together all of that evidence, and we do this in an automated way using the application programming interfaces to pull in new research as it emerges. We then use a pipeline of automated tools to organize this evidence in a way that's useful, tagging it with disease model, intervention, outcome measure, and then we visualize it in a web application that's accessible for everyone to use. And we think this has the potential to change the way we view, conduct, and evaluate research. How does it work? So this is a sort of animation that will hopefully work as intended to just show you how this actually works day to day. So we typically pull from Web of Science, PubMed, and Scopus because they have quite good APIs. And we run a search across these databases on a weekly basis across different diseases. So we'll use the example of Alzheimer's disease because that is the SOLES that I'm going to show you today. So we run a search across those databases and we pull in the relevant studies. We then remove any duplicate studies that exist across the databases using an automated tool called Assist. We then end up with all of our included studies, and what we do is we take a sample of these and send them off to our systematic review facility platform. And this is the platform that we use for collaborative systematic reviews. It allows lots of different people to work on a project at once. So we take a sample, typically a few thousand articles, and we send them off to SURF, our systematic review facility, and we have a team of reviewers essentially screen those articles manually. So they'll determine this is relevant for this study or it's not relevant for this study based on the title and abstract. Once we have some human screening decisions, we can then train a machine learning algorithm, and this is currently hosted with our collaborators at University College London in the Epicenter. So this machine algorithm then allows us to, if it performs well enough, we can then actually apply that machine algorithm to the rest of the data set. So on all of those studies that we've retrieved and all of the studies that are coming into the pipeline on a weekly basis, we can screen out the relevant ones. Once we have our relevant articles, we then download the full text, and there's a number of tools to do this in an automated way according to our institutional subscriptions. Once we have the full text, we then apply a range of different text mining tools to extract information from those texts. So we pull out, for example, mentions of the outcome measures that are used in that article, the models that are used, and here you can see I've got these are some transgenic Alzheimer's disease models as an example. And we also pull out mentions of the intervention or drugs tested in those articles as well. We then use a natural language processing tool to extract information about whether the animals were randomized to groups, whether the experimenters were blinded when they conducted the assessments, and whether a sample size calculation was performed. We also are able to make use of existing tools out there. There's a tool called OddPub, for example, that allows you to extract information on the transparency of that work. So whether the code and data are available or whether there's a statement saying that it's available in the full text. So we can pull out all of this information. And what it really leaves us with is a database of curated evidence. And adding to that, we can pull in additional metadata or information about published work from the CROSREF database and the OpenAlex database. These are both databases of millions of citations in the literature that give us additional information. For example, OpenAlex contains information on whether a study is open access or not, and what the funding information for that study is. We bring together all of this information into what we call an SQL database. So essentially, this is a structured database of all of our studies and the relevant information attached to those studies. And then that sits behind an interactive web application that we make currently using R programming language and R Shiny. So all of this really is actually in an R package now. So all of these automated functions that take you from the systematic search through to retrieving the full text, through to screening the articles and extracting all the information from the articles, we have a pipeline in R to help us do this that we've developed over a number of years now. And we've actually just made this openly available on GitHub. So if anyone is interested in the automation side of things and wants to understand the code a bit more, I would encourage you to have a look at this GitHub repository for anyone that's interested. So in there, we have all of the functions that essentially allow us to run this on a weekly basis for our SOLES projects. What it actually looks like every week when we run this is for an individual SOLES project, we basically get an email update informing us that this script has run or that our workflow has run. And it will tell us how many new studies have came into the pipeline, how many have been screened, how many have been tagged with these different things that I was mentioning. So how many have been tagged by animal model or intervention or species and all of these things. And then every week the applications are actually redeployed with this new information, so they're kept up to date with the latest literature that's emerged. So when I was preparing this talk, I was trying to find some images of what a SOLES might look like without actually just showing you what it looks like. I was just interested to see what ChatGPT could tell me about what a SOLES was based on my description. So based on a lot of the things that I said in this talk, this is what ChatGPT thought a SOLES project looked like, which actually isn't a million miles away from our work in the SOLES development team trying to build a dashboard or a platform like SOLES. Here was another illustration of what it could look like. I'm not sure why I don't know why there's a person here, and I don't know why the person has wings. But now I can show you what a SOLES actually looks like to give you a bit more context. So I'm just going to go to this web address here. Let me just see if I can load this up in the background. You can also go onto this website for yourself if you're interested. So I'm just going to show you briefly what the Alzheimer's disease SOLES project looks like, because this is one of our most developed projects. And this contains evidence from animal and cell-based models of Alzheimer's disease. So this is a platform. As you can see, it's got a bit of description about what we're trying to do. And then if I go to the data collection section, you can see the number of included studies. So the SOLES are updated every week. You can see there's been 56 new studies published in this area this week that have made it into the SOLES pipeline. And in total, I have 41,000 articles that have been indexed within AD SOLES. So this is estimating the 41,000 articles describing animal experiments in Alzheimer's disease models. You can see the number of publications over time is just absolutely skyrocketing. There are so many, over 6,000 in 2023. You can see the need for this. We've also got some illustrations of how many studies have been retracted, but this is still an ongoing process. Keep track of this as well. We've got a bit of information as well on the progress of all of the tagging I was talking about. So how many studies have actually been tagged by the risk of bias measurement, how many have been annotated for disease model, for example, out of the whole data set. And there's also a bit of description in here about the technical details of how we actually find publications, screen them, download the full text, and tag some of these things that I've been talking about. So if you're interested, you can look at this in more detail. We have a page on the transparency of evidence in this space, so you can see the percentage of open access publications, the percentage of publications with open data and open code. As you can see, sharing code in this literature is still not very prevalent, unfortunately. Neither is sharing data. And we can also have a look at this over time to see if there's any improvements in open access, open data availability, and open code availability. We also measure risk of bias reporting, as I mentioned, so you can look at the percentage of studies that have reported blinded outcome assessment, conflict of interest statements, etc. And you can look at this over time as well to get an idea if that's improving. And as you can see, for things like blinding, it definitely is improving over time, which is good. Then we have a page on different therapeutics that have been evaluated in these animal models. So we have a summary of all of the interventions here. So if we look at maybe amyloid-related drugs or drugs that target amyloid in some way, you can see if I hover over here, this is the number of publications indexed in AD SOLES that mention these drugs, just to give you an idea of what the literature looks like. We also have trends over time, so you can look at the number of, for example, amyloid-related therapies over time that have been mentioned in publications and see if there's a trend for certain new drugs, for example, being mentioned more recently. So if we look at aducanumab as an example, this is one of the more recently developed Alzheimer's drugs that has been approved by the FDA. You can see the number of publications there over time have been increasing, which isn't a surprise. Similarly, for different outcome measures, we don't have every single outcome that is mentioned in publications. This is something we want to expand across all the SOLES projects, but we do have quite good coverage of a lot of the neurobehavioural outcome measures and an estimate of how many studies have looked at these. So open field test, for example, or Morris-Water maze. Again, you can look at the trends in different outcomes being measured over time. We also have a similar plot for animal models. We've got different transgenic models, induced Alzheimer's models, and again, you can hover over and see the rough numbers of publications that mention these models. On this page, we have a matrix plot that essentially is trying to map a couple of things together. So to give you an estimate of how many studies have looked at this transgenic model, APP23, and looked at their behaviour in the open field test. And this is giving an estimate of 49 as an example. And you can do this for different models, interventions, and outcome measure combinations. One of the most useful pages is the search page here. So you can see we've got essentially all of the articles in AD SOLES, so over 41,000 are in this table. You can have a look through. You can also search using keywords. So you could look for example, inflammation. If you're interested in studies that mentions inflammation in this space, you could look at that. It takes a little while for the searches to run. You can also filter the studies. So you could filter for, if you just wanted studies published since 2021, for example, you can look at studies that mention specific drugs, outcome measures, models, and then you can apply those filters and you can actually download a reference list. And this is really important because the whole idea is that this could be used as a starting point for a more detailed systematic review if you want it to be used for that. So you can download it in EndNote format, for example, and then this is a starting point of relevant studies. Or even if you're working in the lab and you don't want to do a systematic review, this can still be helpful to help surface research that you might have otherwise missed that is relevant to your research question or your interest. I'll stop sharing. Oh no, I didn't mean stop sharing. I meant to just stop sharing that screen. Let me go back. So I want to go back to the presentation now. And again, it likes to do this every time. So now you've seen what it actually looks like. This is what these platforms look like. I just want to talk about how this can actually help systematic reviewers. And I've touched on this a little bit already. But SOLES really are there to provide a broad summary of evidence in a given research space. It gives you an idea of the quantity of evidence, looking at different models, different interventions, and different outcome measures. It gives you an idea of the transparency of that evidence and a bit of insight into the quality as well, because we measure the reporting of measures to reduce the risk of bias. The idea is also that they can help accelerate and support the conduct of systematic reviews and hopefully reduce barriers to actually starting a review. If you're just getting started with conducting reviews, it can be really daunting to figure out exactly how to search different databases and come up with exactly the right search terms to find the studies that you want. And in some cases, if a SOLES already exists in that research domain, it would be great if people could use that resource as an accelerated starting point and just download their systematic search from there, knowing that it already has come from at least 3 databases, so PubMed, Web of Science, and Scopus, and been through a deduplication and screening process, so that it's hopefully more relevant. So you're going to end up with less studies to go through and hopefully save yourself a bit of time as well. And also the idea is that we're sharing the benefits of this automation. So not everyone is up to speed on all of these automation tools. Not everyone feels comfortable in applying them or coding in R or Python. So we wanted to open that up, open up the benefits of that to a wider audience. We also wanted to use SOLES for community building and collaboration. I think there's opportunities within these SOLES projects to start reviews from the same starting point within a research domain, and opportunities for coordination as well. Going beyond systematic reviews, we hope that SOLES can surface evidence for research users. So as I mentioned, if you're working on a project in a lab, and especially if you're working in Alzheimer's disease, for example, you might want to go onto this platform and use it to see if you've missed any relevant studies out there that could be useful for you. It also, in the long run, hopefully will help to avoid research waste. So if you're able to go on here and check if someone's already done that experiment, it might help to reduce the same experiment being done again unnecessarily. We also want it to be used as a tool to help inform funders and policymakers to see how much evidence exists already, and perhaps to identify how feasible another project might be and how much there is already in that space. It can also be a tool to share research with patients and the public, because it's an openly available resource for people to go on and look at the literature in a given area. We also think it could be a tool to help inform research improvement activities. So as you've seen on there, we can track things like reporting quality and transparency over time and see if it's improving. And we can look across different fields as well and see if there's a difference. So we actually now have over 10 SOLES projects that are either fully established or in progress. So I showed you the one in Alzheimer's disease, but we also have one in depression, psychosis, neuropathic pain, neurodevelopmental conditions, stroke, the impacts of pesticides and other areas as well in the pipeline. We have just literally, like yesterday, created a website here that you can go to that just showcases all of these SOLES projects and puts them in one place, because quite often they were sort of hidden away and you had to know to contact the right person. So we're trying to make it easier to make it clear what's available already and what domains have been covered. So feel free to have a look at this website and navigate to any of these. I want to emphasize that all of the projects actually have a shared underlying architecture. So as I mentioned, they all have this SQL or structured query language database. There's a database for each of the SOLES projects, but actually it's the same. And the idea is that it means that it's actually quite easy to combine different SOLES projects. And that's something that we're actually working towards in the near future. This, obviously, you won't be able to actually read or understand all of this, but this is just to show you what the databases actually look like and some of the tables that we have in there, sort of behind the hood of a SOLES project. Each study has a unique identifier, and that's mapped across the different tables where we extract different information. So why would we ever want to combine SOLES projects? This is something that we're quite interested in, because we're finding, as we've done more systematic reviews over time, what we find in the animal literature is that actually sometimes the same models are used across different diseases. And sometimes taking this disease-centric approach is not that helpful. And we might be missing latent connections between different spaces. So this is an example in the Alzheimer's disease space that was essentially giving rats STZ or streptozotocin. I always really struggle to say that for some reason. So I'm just going to say STZ, which is an inflammatory intervention. And this can lead to inflammation in the brain, hence why it's seen as an Alzheimer's disease model in some cases. We also found in the neuropathic pain literature that STZ was being given to induce diabetic-induced neuropathic pain. In both of these examples, in completely different literatures, they're actually giving the same model induction, STZ, and they're measuring it on the same outcome measure, which in this case was the open field test. Under normal circumstances, we would never look at these together. This data would never be combined, because you typically do a very disease-focused search. When you're searching PubMed, for example, you would only look for Alzheimer's disease or neuropathic pain. So that's why we're interested in actually looking across different SOLES projects and seeing what the overlap is. So we've started to do this with a meta-SOLES that's combining these different projects. And we had a look across the Alzheimer's disease literature, the stroke literature, and the literature on pesticides or plant protection products. And what we're already seeing is that there's actually a huge overlap in terms of the compounds that have been investigated in these different fields and in the gene targets that those compounds are actually influencing. So this is very early work, but we're essentially even more surprised at the amount of overlap here, and we think there's a real potential to unearth some new biological insights from combining this information. So moving towards the future, we really want to work with people who are interested in conducting SOLES-derived systematic reviews. So if you're working in a certain research space and you would like to use SOLES as a starting point, please reach out to us. And you can use that website now to contact the relevant person who's in charge of each SOLES project. We're interested in trying to see how this approach compares to traditional systematic review approaches, to see whether it is more sensitive, so can pick up more relevant, more of the relevant literature, and also how much time it can save. We're interested in trying to improve the automation side of things. So at the moment, we're really just looking in the full text for mentions of different animal models, different treatments, and different outcome measures. And we want to make this a lot more specific, because sometimes an animal model might be mentioned in reference to a previous publication and not actually evaluated in that study. So we want to refine those methods and continue to make them better. We also want to better deal with research trends and shifts. So, for example, the machine classifiers that we're using, how do we know when they're becoming out of date and when the field has moved on? So we need to work on methods to improve that. We want to, in general, make sure that these SOLES platforms are fast. Sometimes there's a bit of a delay when you first go onto it, so we're working on speed and efficiency gains. And we also want to expand into different types of data. So most of the SOLES that I've shown today have data from published animal research, but we want to also expand this to include in vitro research and clinical research and omics data as well in the near future. So that's my last slide. I just want to say thank you to the CAMARADES group here and across the world, our collaborators, all of our funders for this work, and a special thanks to the SOLES development team who've made all this work possible.